Good afternoon, ladies and gentlemen. Thank you for coming to this pair distinguished lecture today. And today we are very honored to have Professor Chang and also our Deputy President, Professor Wang, with us. So many of you might already attend other pair distinguished lectures, but for those here first time, I would like to introduce this pair distinguished lectures series. We organize this at least once every month, and we always invite high-profile scientists, engineers, and university administrators to give these distinguished lectures. And at the pair, we have conducted research in three major areas, like sustainable cities, advanced technologies, good health, and well-being. So today's lecture belongs to the last category. In this last categories, at the pair, we have 7 research institutes and research centers working on that. And today's event is sponsored by the Research Institute for Future Food, we call RiFood. And in addition to this, in the same area, we also have the RI on smart aging, Chinese medicine innovation, sharp vision, sport science and technology, mental health, and also the newly established translational technology for tourism. So I hope that you will enjoy the lecture today. And now I will pass the microphone to Professor Wang, who will introduce our speaker. Thank you.
Okay, good afternoon, colleagues, friends, and students. So it's my pleasure and honor to be here and to introduce our distinguished speaker today, Professor Eugene Chang, okay, from the University of Chicago. So Professor Chang is a physician scientist, okay, so he's the Martin Brewer Distinguished Professor of Medicine at Chicago Knapp Center for Biomedical Discovery and the Director of Microbiome Medicine Programs. And he also recently served as the Senior Fellow of PAIR. Professor Chang's research focus is to study the host, microbes interaction and disease mechanisms that underlie complex immune disorder, including IBD, okay, and also the metabolic disorder such as obesity, metabolic syndromes. Many of these studies are supported by national grants and involve collaborations from colleagues from all over the world. So Professor Chang is committed to translate his research into the health solutions. He's the co-founder and the chief medical officer of a company, AB Neuron, okay, therapeutics. It's a company dedicated to develop a new class of microbial peptide that maintain healthy fungal microbiome by preventing virulence and infections. He's also one of the key collaborators of Microbiota I Center. Okay, it's an Asian-based microbiome innovation center founded by the Chinese U. Okay, so I'm sure many of you have already heard a product called Genie, right? So basically, it's a product that's developed by the CHK spin-off company. So Professor Chang received his medical degree doctor from University of Chicago and has been contributing professionally to his field for more than 30 years. Okay, so you know what, Professor Chang is a really productive scientist. Since 1990, Professor Chang has published more than 500 research papers. Scary. Okay, so and is also a highly cited researcher for microbiology in Journal 23. And amongst the most many prestigious honors, Professor Chang was awarded the National Institute of Health Merit Award for 10 consecutive years. Oh, it's amazing. So, and also include the Takata Distinguished Scientist Award and also Outstanding Mentorship Award by American Gastroenterology Association. Oh, the many is a lot. Okay, I'm not going into details. Otherwise, I need more, 15 minutes more. Okay, so without further ado, okay, so ladies and gentlemen, colleagues and friends, please join me to welcome Professor Eugene Chang to the stage. Thank you.
It's a great honor to be here. And I'm particularly honored to be appointed a senior fellow of PAIR. This has so far been a promise that, we would interact and collaborate. But I think from discussions this morning that I've had with colleagues here at PolyU, this promise will become a reality. So I'm going to talk about a question that I think remains unanswered, and that is, what is a healthy gut microbiome? And I'm going to provide a reason or reasons why it is really important to know. I think that the answer to this question really will open the door to many new ventures, particularly in the area of microbiome-based interventions. So let me begin by first a disclosure. So I am the founder and chief scientific officer of a company called Gateway Biome. And some of what I'm going to talk about today is relevant because it was developed through Gateway Biome. So let's start with the question of what is a gut microbiome? So the gut microbiome is more than just the microbes in the gastrointestinal tract. It's the totality of microbes, and it includes all the genetic elements and the environmental drivers that form this ecosystem. And just some fun facts that I'm sure many of you know about the gut microbiome. I always view the gut microbiome as a vital organ of our body. It fits the definition of a vital organ. We cannot live without it. It provides numerous benefits. And it is something that, like any other organ system, it can go bad. And when it goes bad, it can cause disease. Furthermore, like other vital organs, for example, the liver or the heart, you can transplant it from one individual to another. We call that fecal microbial transplant. But the same principle. We take somebody's vital microbial organ and we can transplant it into a recipient. And like a vital organ transplant, if there is not a good match, parts of that vital organ will be rejected. So the other fun facts are that this is really the largest vital organ of the body if you consider it on a cell basis. There are at least 10 to 40 million trillion microbes in the GI tract. That's 10 times the number of cells in our entire body. And not surprisingly, if you look at the number of genes that are present in this vital organ, which is estimated to be about 3.3 million genes, this really dwarfs our own genome. It's our second genome, but it's the largest genome that we have in our body. If you look at the weight, it's about 2 kilograms. And if you compare that to other vital organs, for example, the heart or the brain or even the intestine, it's a fairly large organ. And then another important fact is that the cells or the gut microbes that occupy the GI tract are very, very host specific. And it's determined by drivers that come from evolution and co-speciation. What I mean by that is that if you look at the microbiome, let's say of a bird or a mouse, those microbes you'll rarely ever find in a human gut, and vice versa. You will rarely find human gut microbes in other species. Now, what does a gut microbiome look like? Well, that's shown in the figure on the right. And It's shown here, this sort of amorphous mass that lies on top of this mucus barrier. We are below here in the lower left. This is the colon. And there are two parts of the gut microbiome. There's the part that's attached to the mucus surface, and then there's a part that we call planktonic, which are free-floating gut microbes. So the other thing I should mention is that, often we talk about the gut microbiome, but actually we should be talking about the gut microbiomes, the plural form, because the GI tract is essentially a number of ecosystems that are created and are connected in series. And each part of those ecosystems are different. We evolve these ecosystems to attract and engraft specific microbes. So it's not a random process. It's a specific set of microbes that provide essential functions and benefits that we must have. So what do these gut microbes do for us? Well, this is something that I think has become evident over the past couple of decades. They do numerous things, not only within the GI tract, but also systemically. Examples are that it provides a barrier to microbes that would otherwise enter the bloodstream. It provides resistance to pathogens. It is important for the development and the regulation of immunity. And it's also essential for digestion of dietary components and their absorption. But like any other organ of the body, things can go wrong. And when these perturbations or imbalances occur in the gut microbiome, otherwise called dysbiosis, there are many diseases that can result. The ones that I'm interested in, for example, inflammatory bowel diseases and certain forms of liver diseases, particularly fatty liver disease, clearly have a microbial basis. And we're just trying to understand what that basis is. What is the driver? What are changes that are a consequence of the disease that has been promoted? But if you look at other types of diseases, including heart disease, cancer, respiratory diseases, et cetera, there are numerous reports that suggest or even show that gut microbes have a role in either the causation or the pathogenesis of these disorders. So I can come back to this question that we began with. How do you define a healthy gut microbiome? Now that might seem very intuitively obvious, but actually this is the main question that has been unresolved. And why is that important? I think it's important because if we can define whether a microbiome is healthy, we will be able to define an unhealthy gut microbiome or dysbiosis. Now, you can read in the literature about how people have defined a healthy or unhealthy gut microbiome, but by and large, I think most have missed the mark. And why is this important? Well, if you look about, on the television and the news, research reports, there's a lot of information about what the microbiome or microbiome-based interventions actually do. But if you look at the science behind it, there's very little evidence-based data that show that these interventions actually work. And this includes things like prebiotics, postbiotics, probiotics, phage therapies, microbiome transplantation, either undefined or defined. And I think that this is a major stumbling block because we're actually developing these microbiome-based interventions based off of assumptions and anecdotes. We really have no scientific evidence whether these things are actually working. Yet we have multi-billion-dollar industries that are promoting these products as being either beneficial to health or for treatment of disease. Yet there's no scientific basis to support any of their claims. So this has led to a recent commentary that was published in Science, and I'm going to take a minute just to read through it, because I think this really crystallizes the problem. So this is an article that appeared just a couple of months ago. So the abstract reads, a growing body of research has suggested the potential for improving human health by understanding or better understanding the human microbiome. So this has led to the emergence of a global industry that sells direct to consumers. But it also has been a industry where science has been ignored. And many companies have made claims of having the ability to detect abnormal microbiomes, but they're not substantiated by research, and therefore lack analytic validity, and the results have no demonstrated clinical ability. So it concludes by saying, to address these concerns over potential harm, we conclude that regulators should develop requirements for the industry to document and demonstrate the consistency and validity of methods and claims. So I believe that this is actually crystallizes for me what the problem is and what we have to do. Okay, so let us step back for a second. There are other vital organs of the body. Some are shown here: brain, heart, kidney, liver. In every case, there is a diagnostic. There are a set of clinical tests that will define the state of health or unhealth of these vital organs. For example, if you take the heart, we have electrocardiograms. We also have serum tests like cardiac enzymes that will tell us whether the heart is working fine or if it's having trouble. Now, on the right, I show a screenshot of my own complete blood count. Okay, so what's really vital to clinical tests are these normal reference ranges. So that's indicated by the green bar. And fortunately, most of I think all of my indices are within that reference range. So it makes me happy. Now, if something were out of that range, then I know that as a physician and also as a patient, that I need to find out why, right? So this is why these clinical tests are so important. Now, for the gut microbiome, what do we have? We really don't have very much. What has been used in the past is a number of non-cultivation tools, which I'm going to review very briefly for you. So the most commonly used tool is a tool that we basically amplify a certain region or regions of the gene that encodes for the 16S ribosomal RNA. And this has been very helpful. This was a big breakthrough. The technology is now very cheap, very fast. Almost anybody can do it. But the problems with 16S is that if you look at people's 16S profiles in this room, everybody is different. So how can you possibly determine what is healthy from 16S? all it tells you is who is there. It tells you nothing about the function of those gut microbes. And if there's no normal, everybody is different, there's no way to define a normal reference range. So this is very problematic. And I think 16S amplicons fall short of defining a gut microbiome, despite whatever other people have said, trying to assign function to certain taxonomical groups. I don't think that really works. So then comes along shotgun metagenomics. This is where now we have the ability to sequence every microbial gene in a sample. This was a big breakthrough, but the problem with this is that it provides too much information. You get thousands and thousands of sequences. And yes, you can identify maybe 20, 30% and be able to define them in terms of functional categories. But the problem with that interpretation is now you're looking only in a subset of those sequences. You don't know what the rest of the 70 or 80% actually is telling you. So you could be skewing your interpretation. Now, the one thing that really has helped with shotgun metagenomics is that shotgun metagenomics, unlike 16S ribosomal RNA, can get you down to a strain level. 16S gets you down to genus level at best. And that to us as researchers is very important because we can now deep sequence and put together nearly complete genomes of particular strains just by contig assembly. And that has allowed us to do a lot of things, from just identifying pure strains, understanding the synteny or the order of the genes in a circular genome, but also to be able to do some genetic engineering because we now have a map. But as a clinical tool, it really is too costly, takes too much time, requires a lot of expertise, just, I think, is not clinically practical. And that goes for metatranscriptomes as well. So metatranscriptomes is shotgun sequencing and reverse transcribing messenger RNA. There are a lot of problems, a lot of difficulty in seeing the messenger RNA because most of the sample is structural RNA, that is either transfer or ribosomal RNA. And then there's metabolomics. And I'm going to just say that this is a measure that my lab currently uses because it's a direct measure of function. And function tells you much more about what the microbiome is doing for us and what interventions, let's say like probiotics or FMT, are doing to the gut microbiome. So I'll get back to that in a little bit. I just want to say that like metagenomics, metabolomics, which is done by mass spectrometry, is also very expensive, takes a long time to turn around, and in its current form, it's very difficult to use, and I don't think it has real clinical application. Okay, so let's look at some other features of these tests that we have to try to assess the state of the gut microbiome. 16S, as I mentioned before, there's really, if you look at the top panel there, and these are basically membership of the microbiome at I think a phylum or class level. Every row is an individual. This was part of the Human Microbiome Project of the NIH. You can see that everyone is healthy, yet the percentage of their taxa are very, very different. If you look on the very far left, You can see the Bacteroidetes, which are in the blue, outnumber the number of firmicutes. This is a very commonly used test, Bacteroides to firmicute ratio. But that person on the right who has a ratio that's less than 1 actually is also healthy. So how can you use 16S to define the health of the gut microbiome? The answer is you can't. Now, if you look at the metagenome, again, individuals are each column. Here you see something different. You see that there is homogeneity of these functional subsystems. So these are categorized not by the specific gene sequence, but to a functional category to which they are assigned. We're pretty much the same. Now that's very important because now you have a metric that comes closer to what might define normal. But again, remember, metagenomics is DNA. So you're looking at functional capacity. You're not actually looking at function. You don't know which genes are actually being expressed, right? So it's helpful, but again, it's costly, it's difficult to analyze, turnaround time is slow. So it's not very clinically useful. Okay, another technique that is commonly used is taxon calling or Bacteroides to firmicute ratios to define a dysbiosis. And the problem here is it assumes that microbes are either good or bad. And that just is such a oversimplification of what microbes actually do or how they function in a specific context. And in many of our studies, for instance, we find that a good microbe can turn into a bad microbe based on the context and whatever stress environment it encounters. And the way that these microbes can mutate or acquire genetic information is just mind-boggling. There's so many ways that they can transform from one type or function to another. And so this, to me, is very important to recognize because an E. coli can be very helpful and beneficial to us in one circumstance, but if any of the E. coli ever got into our bloodstream, it becomes a pathogen, right? Okay, so let's turn to the question, what are desirable features for the assessment of gut microbiome health? Okay, first, let's try to develop something that's non-invasive, maybe from stool or blood samples. It should be a panel that is representative of the different important and essential functional subsystems that the microbiome provides to us. It should be quantitative rather than relative. You know, metagenomics, 16S, most of these are all relative values, percent of abundance, for example. And what we need is something that is absolute. The blood glucose is a good example, right? We have normal ranges for blood glucose. It's very quantitative, and you know whether you're in the normal range or outside of it. And then metrics really need to have a normal reference range. And I think this is why you have to make the assay quantitative. And has to have reasonable costs and turnaround time. Home collection is much better and easier for the patient or subject. It's very hard for an individual to produce a stool sample on demand. And I think that this gets around the problem that we face with trying to collect these samples in the clinic. And then there's the issue of reproducibility. So how do we get around these problems, and how do we get a a tool that fulfills all these criteria. So I'm going to make a case that this can be done through an approach that is based off of metabolomics. Why metabolomics? Well, if you think about it, metabolites are the final product. They are a direct reflection of microbiome function. And if you look in the left panel, you can see that, for example, there are many types of microbes that are in group 1, but they all make metabolite X. In group 2, there are many different types of microbes that make metabolite Y. But if it's the metabolites that are the actual effector molecules that we benefit from, or in some cases may cause disease, this is what we want to measure. Do we really care who makes metabolite X or Y? I would argue no. All that is important is knowing what the levels are of metabolites X and Y. All right? So that's the first criteria. Now, metabolomics right now are done by mass spectrometry. It's a very sophisticated piece of instrument. You get 10,000 features or signals in one sample run. That's just too much. We can't even recognize 2/3 of those features. And this is why it's not useful. So we ask the question, do you need all 10,000 features to be able to define the health of a gut microbiome? Can we get away with a more manageable number? What about 20? Does 20 work? Does it work as well as 10,000 features, particularly if we know what those metabolites are? So we did a approach that was partly supervised. That is, we determined that there's certain markers that we would like and we think are highly representative of specific functions of the gut microbiome. And we did apply machine learning and artificial intelligence to determine which of those markers would perform best in representing that particular functional subsystem. And then we determined, can we get away with 20 that will be highly informative and perform just as well as 10,000 unidentified features? And the answer is, we figured it out, otherwise I wouldn't be here. And basically what we've been able to do is use a discovery cohort, which is about 85 healthy subjects, adult subjects, and we defined 20 metabolite markers that represent different functional subsystems of the gut microbiome that are fairly homogeneous from one healthy individual to another. And that way we developed a reference range. We also developed an approach where the mass spec, from the time that the sample is collected to the time that it's analyzed, was rigorously quantitative. So we used internal standards, no more relative values. This was absolute value. And by doing so, we developed a quantitative tool that now can provide not only an assessment of whether you have a eubiosis or a healthy microbiome, or a dysbiosis or an unhealthy microbiome. And more than that, this panel pinpoints what the problem is. For example, if you have a dysbiosis, you'll find out, for example, in a couple of use cases I'm going to show you, that you can determine, for example, if an individual's fermentation is normal or it's awry. So, but there's a practical consideration. People don't like collecting stool. They hate it when, they collect a stool and then they have to stick it in their freezer. It's just a big yuck factor. And so can we get around this issue? And can we get reproducibility? And what I mean by reproducibility is that if you let a sample sit for more than, let's say, half an hour, microbes are metabolizing. They're changing the metabolome all during that time. So you have to develop a way to stop microbial metabolism and be able to establish procedures that allow you to accurately quantitate that metabolite at the time of collection. So this is what we did. We developed a home collection kit. And the home collection kit provides a way to make sure that there's an adequate biomass, but also contains certain internal standards that we use for quantitative measurements of these metabolites by mass spectrometry. Moreover, this preservative has the ability to extract certain types of metabolites that we're interested in so that we get reproducibility. All right, so does it work? All right, so this patient, we call him patient Q, came to me about 3 years ago now with a history of what we call functional bowel disease. This started 10 years ago when he was placed on 18 months of antibiotics for unclear indications. And then he started developing symptoms, very incapacitating symptoms that included things like incontinence, stomach pain, increased food restriction or intolerance, and chronic GI discomfort. He went to 4 major medical centers. I won't name which ones, but there are very reputable medical centers in the United States. And they couldn't find anything. He had normal blood tests, normal colonoscopy. So then he contacts me and he asks, well, could it be my microbiome? And I said, I don't know, but we just developed a test that maybe can determine whether you have a microbial basis for your functional bowel disease. So this patient, we've been collecting stool samples 3 times a week for now almost 2 years. And we found that, as I will show you, that by our panel, he had a very severe dysbiosis. He was missing major taxa that we should normally have. He didn't have them. So we worked with a dietitian to gradually increase through a dietary intervention program where we added more and more food groups, starting with a low FODMAP diet. This is a low fructose oligosaccharide diet. Then we started adding more to it. And we used the tool to guide our decisions and management of this patient. At the same time, we also monitored his 16S and metabolomics. So this is just a rough outline over the first 50 months. And the colored squares at the top indicate the kind of symptoms he was having in that particular point in time. And then we, right below those squares, are the kinds of dietary interventions we began to introduce. And over time, as you can see in B, although it's not quantitated yet, but we, at the time that I put this presentation together, we saw a gradual improvement of his symptoms. Now, this is what his metrics showed. All right, let's start with the right panel. So the right panel actually shows an overall metabolomic score. It's a Z-score that we derive from the averages of deviation, standard deviation from the norm of our reference standards for that particular metabolite. And he started with a score, I think, about 42, which is really abnormal. This is way off the curve. 20 is probably borderline. Below 10 is clearly normal, right? So he was 42, and then we started his dietary intervention. And as you can see, he had progressive decline in his score, which translates into improvement in the state of health of his gut microbiome. So that by week 34 to 41, he was actually normal. He had a score of 5. That was well below 10. So what happens with the 16S? Well, you can see that in panel A. Here, there's no change in the Shannon Diversity Index over that entire period. Now, what this means and how we interpret it is that we didn't do anything about what kinds of microbes he had. We worked with the microbes that he had from the very beginning. We were just getting more out of the microbes that he had through dietary intervention. We were able to correct his dysbiosis and put it into functional eubiosis, but we did not change his membership. Okay, so the next phase, which we're doing right now, and I don't have the data for you, is that we're trying to increase the diversity, put back some of these microbes that he's missing so that we can buffer. He has now not only stability, but he has more resilience in case of any perturbation. Now, another key thing about this is that when you look at his scores, we began to see significant improvement by day 90. And by day 1, and he was still symptomatic. But by day 180, as his score got lower and lower, he began to feel better and better. So these changes occurred much earlier than symptomatic improvement, and in many ways gave us hope that we were on the right track in terms of his therapeutic program. I'm going to skip this. Okay, I wanted to talk about one other thing, and that is we've been working with a group. They have a program called Good Food is Good Medicine. And this is an educational program where they go into underserved, underprivileged areas in the Chicago neighborhood where there's higher than normal incidence of metabolic disorders like fatty liver disease, obesity, diabetes, because these are areas where there are food swamps, food deserts, a lot of healthcare disparity. Most of these patients don't have the insurance for these new types of incretins that are very effective in controlling metabolism and in treating obesity. So we tried this program. And the question was, can we show objective evidence that this program is actually working? And if you look at panel A, I think the evidence is yes. Now, this is just a six-week cohort. And you can see that before and after, there is a significant decline, particularly in certain metabolites that we found to be out of the range. And that's shown by the Z-score in the upper panel on the right. And individual scores are shown in panel B. The bottom line is that this tool allowed us to not only say that this program actually does have effect, but when we showed individual subjects their results, they became highly motivated because they could see objective evidence that this program was benefiting them. And they stuck with the program. They re-signed into the class. And they're continuing this program, and we're continuing to monitor their metabolome. Okay, so how do we use this in the future? Well, the way I look at it is that there are at least 3 types of usage. One is that we can monitor a healthy microbiome, make sure that it stays in health. That's in the panel on the left. We can use it to determine the efficacy of treatment. That's shown in the middle. This would be patient Q. Okay, but also it could be used as outcome prediction. That is, response to therapy. Our biologics that we're using in patients with IBD were actually working before we wait 6 months later for a colonoscopy to determine if there's endoscopic healing. So you save a lot of time, a lot of money. Okay, now, I think this panel works great for adults. However, if you look at the ends of the life spectrum, either early life or late life, things are different. There, the reference ranges don't work because the early life microbiome is constantly developing. Things change, the reference ranges will change. Same goes for the late life microbiome. But if we could develop reference trajectories, just like we have trajectories for anthropometric measures like height and weight that pediatricians use to ensure that development is proceeding along well, we can develop these reference trajectories. So on the left, if something happens, let's say they were either a C-section or they were put on formula feedings or eating bad diet, antibiotics, and they're off trajectory. We know we have to intervene at that point, and we can restore that trajectory through a number of measures that can bring back the microbiome to eubiosis. This is really important in early life because the development of your immune system and metabolic systems are highly dependent on the normal development of your early life microbiome. And the same goes for the late life microbiome. And I'm going to skip this part and just end with, as I looked into RI foods, it dawned on me that the vision and the mission of RI foods is very compatible with what we're interested in doing. In fact, I think that we are a very good complement. For the goals that you're trying to achieve, we have a metric that can tell you whether or not your interventions, your probiotics, your whatever you develop in terms of, let's say, medical or prescriptive foods has a benefit. And it will tell you exactly what it's doing to the gut microbiome. And I think that this is exciting for me. I think it was very exciting when I talked to Ka-hing and also to Amber this morning. We had a great discussion, and we're going to do something about this. We're going to work together and hopefully, establish a reference laboratory at PolyU where we can start gathering data in a healthy population, but also in a population where there may be dysbiosis. And that may lead to more effective and therapies and interventions that are evidence-based. So I want to end because this work is really through teamwork. Everything I do is through teamwork. I recognize that I do not have the expertise that is necessary to answer these questions. So I depend on the team of experts. These are people with different backgrounds, but they are the heavy lifters of everything that I presented this morning. So it's important to recognize them. And I will stop there. And I guess we go on to the panel discussion. Anyway, thank you so much.
Thank you, Jean, Professor Chen, for giving us a really excited talk about this. As part of the member of the RiFood who collaborate with the other members who are actually present today, I believe we have, some questions from the audience. So maybe may we invite the questions from the floor first? Yes.
Thank you. So I'll start with the easy one first. And so Professor Chen, thank you for the exciting talk. It's really inspiring. So you're kind of building the standard in the field, and that's that has been like a long question in the field. I'm wondering that your metabolite score, does it fit to the people at different gender, different age, and different races?
That is an excellent question. Okay, I don't know the answer yet, but this is something that I want to work with the PolyU group, because one of the first questions we want to know is, are these the selection of these markers and the development of a cumulative score different for, let's say, a Pan-Asian population versus a North American population? I don't know the answer. I'm hoping that with your help, we'll figure that out. The second thing is that, yes, there are gender differences, okay? Not for all of the markers. There's certain markers, I think like 2 markers that are very different in reference range for male versus female. So these are questions that I think we have to answer. We need help because it's hard for me to recruit out here. I need help. And I think this is a perfect opportunity for us to collaborate. That's all.
Okay. So for my group, we have been working on osteoporosis for a long time. There's always differences between female and male animals in terms of the response to different treatment. So recently, we also measured the bile acids profile, and it's completely different in terms of how they respond to different treatment. So I believe if different life stages in terms of like hormonal stages will also affect those numbers. Would that be true?
I think that I don't know the answer, but I'm guessing that there may be, right? Because the microbiome is very, I always call it the canary in the coal mine. You know, so a lot of the changes that we see, which we call dysbiosis, could be an effect rather than a cause. So inflammatory bowel disease is a good example. We typically see this, what I call an inflammation panel. You can see it clearly by 16S. But you see the same profile in experimental colitis. So it's really not specific to human IBD. It's just a result of immune activation and inflammation. But it becomes a very important marker of disease activity. And that's what we've been using it for. I mentioned very briefly that, you know, that we can develop this tool as a biomarker of therapeutic response. So in the IBD field, we start these patients on these biologics. They're very expensive. And frankly, we have no clue why some people respond to a biologic and others do not. If you start a biologic, you know that your response rate will only be 40% or less. And you won't know till 6 months later when you do that colonoscopy whether you had to switch that biologic. By then you spent a lot of money and a lot of complications have developed. So we have a study ongoing now where we're looking at this tool to determine whether or not the therapy is working even weeks after it started. And if it's not showing improvement, then it's time to switch to biologic. And I think that this will reduce both the development of complications as well as the cost. And this can be all monitored as an outpatient basis.
Any questions from the floor? Yes, please.
Yeah, thank you for the talk. You have mentioned several omics, like proteomics, the metabolomics and the transcriptomics. How you combine them? By using the KGG pathway?
I'm sorry, the last part I didn't get. How do we combine them?
Yeah, how you compare the result of them because they are from the different dimensions.
Yes, that's a good question. And this is one that plagued the NIH Human Microbiome Project. It was a second phase of it. You know, how we had all these omics that we were collecting. How do you integrate that data? I'm not sure that it's easy to do that for a variety of reasons. First of all, there's no standard operating procedure when people do like shotgun metagenomics or filtering of that data. Same goes for metabolomics. So you have the problem of reproducibility even among institutions. But the second problem is that they are very different metrics and they tell you different things. Can you combine them to get more power out of your data interpretation? I don't know the answer to that, but I'm guessing that the answer is yes. But to do things like shotgun metagenomics, those of you, I'm sure, that have done it in this room, it takes a while to get through that data. You get a lot of data and you have to sift through it. You cannot do that manually. You have to use algorithms in order to see what is connected and how you can project something meaningful out of this data. So for the moment, I'm sticking with metabolomics because that's something I understand. I know what these metabolites are doing. I can tell you what groups of microbes are actually producing these types of metabolites. And ultimately, I think that unlike any other vital organ of the body, the gut microbial organ is one where you can actually make a change, right? The gut microbiome can be manipulated. It can be rebalanced in many different ways. In the talk that I'm going to give tomorrow at the FSN seminar or salon, yeah. And I'm going to show an example of how you can reshape a microbiome, not by replacing it, but by using ecological and evolutionary principles to rebuild a healthy microbiome from a dysbiosis.
Okay, I actually have a follow-up question for that. You know, like as a person who works on the gut microbiomes, and many of people now are using like germ-free mice to demonstrate that gut microbiota is one of the causative agents to keep leading to many disease. And but I noticed the data, although it's just a snapshot of the intervention you have for this patient queue, these diet interventions are mainly, I just had a quick look. It looks like many of them are actually prebiotics. Is it like many of them are kind of like prebiotics? And the reason using a germ-free is that we are not satisfied as the correlational studies, and you know, like the correlation observation derived from the nutritional epidemiologist. But the end, it looks like the using the biomarkers that you just mentioned, we actually, what would be the next step? What if we have set up the reference range? Then what can we do to improve the markers? Does that mean in the end, we still have to come back by the diet interventions and following the observations we have from the correlation study?
I get asked that question all the time. It's a very good question. So I look at it as, you know, something that is evolving. We just created the first stage. Okay, the ability to determine the health of the gut microbiome was a major obstacle. It really prevented us from developing effective interventions. Now that we have this, we know it works. It may have to be adapted to certain populations or to gender,
Mm.
but that can be done. But now we are able to move to the next stage. And the next stage is developing targeted interventions to in a precision way, correct a dysbiosis. Because the panel tells you exactly what the problem is. So now you're not guessing when you introduce a therapeutic. In the future, I predict, and I'm hoping that this is going to come out of our collaboration, because PolyU is way ahead of the curve in terms of thinking about functional foods or medical foods. I think the future is going to be in developing a suite or a inventory of diet-derived compounds that we, through, let's say, a functional tool like we've developed, be able to know precisely what it does. And now we can use them prescriptively, right? This is what we do in medicine, right? We know that digoxin is, which by the way is plant-derived, and we know what it does. It will strengthen the cardiac function as well as it can treat certain arrhythmias. So we know exactly how to use it, but we had the tool to determine what the problem was, whether it was an arrhythmia or a problem with ejection fraction. Right? We want to do the same thing with this microbiome tool. And I think that we should be able to develop the next generation of medical foods that are used just like we use drugs. We use antibiotics for infection. You know, we have antihypertensives for high blood pressure. Well, this is the same thing. We're going to see that in the future.
Okay, thank you. So we have a question from the online audience. I think this one is more close to the interest of general public. So the question is, Professor Chen, I would like to hear your advice on global human microbiome market. Consumers are adopting a more holistic view of their diet and lifestyle choice with gut health at the center. We are seeing too many microbiome supplements nowadays. So for general consumers, how should we decide which to buy but not the others? That's a question one. And the second question is, what are your views on the current regulation and landscape of the market? The third one, what can be done to ensure quality projects and consumers' decision-making? Well, that's a tough one.
You'll see that my short-term memory is very poor. So the first question, can you repeat the first
For
question?
general consumers, how should we decide what to buy but not others?
So I think that what we, like this commentary that was published in Science, what it says is that we have to make accountable all companies that are developing microbiome-based interventions for their claims, right? And you have to hold them to some scientific evidence that their claims have merit. Only then will you develop trust and confidence in the public that certain interventions will work under with different indications. So I think it starts with a cultural change. What was the second question?
The second question is, what are your views on the current regulation and landscape of the market?
Well, for most microbiome-based interventions, there's no regulation, right? The Food and Drug Administration in the United States, which is a very strict regulatory body, you have to show evidence of efficacy. The probiotic, prebiotic companies basically have bypassed it. So they don't want this, what they're producing or marketing, to be a drug. They call it a nutraceutical, right? So it doesn't fall into any of the regulatory requirements of the FDA. All they have to do, they cannot make any claims of efficacy in treatment of disease. Except in one case, the new FMTs that have been coming out, and 2 of them have been given FDA approval for just recurrent C. diff infection. But all others are considered nutraceuticals. They are marketed as direct to consumer. And I think that the consumer right now has no idea whether or not these things will work. So we have to do better. We have to make these companies accountable for the claims that they make. So what was the third question?
The third question is, what can be done to ensure quality project and consumers' decision-making on the microbiome products, I believe?
Well, so I believe that we had to put a lot more science into the development of these products. So little science is actually applied, or even, I would even say neglected, because most of I hate to say this, but most of these companies are out to make money. They don't really care whether the product works or not. They just want to make it sell. We have to change that culture.
Yeah.
And I think that we have to be rigorous in our getting the scientific evidence that these products actually work, and building trust with the public and doing so.
Yeah, I think public education is also part of this.
Very much so.
Yeah, because not many people will really read the labels of those type of the products. They probably just look at the advertisement or promotions and they would buy it. And simply because they don't have relevant backgrounds or something like that. So I think that could be done in several ways.
I just want to echo one thing, because our department is going to train dietitian. So what would be the role of dietitians in improving the gut health?
I think that diet is the most effective, safest way of changing a microbiome. So I believe that that's the first approach. And in order to manage patients, you need a dietitian. We have a dietitian, research dietitian, who knows exactly what we're doing and works with us. And she, based off of each week's results, she will determine what the next course of action will be. And then there are some instances where the addition of a new food group may cause intolerance or actually worsening of the microbiome index. And so we have a tool that will tell us whether we're on the right track or not. But I do think that dietitians are absolutely essential, because this is really the safest, least costly, and probably the most effective way of correcting a dysbiosis and restoring eubiosis.
We actually have a question related to this one. Where do we find information to diet that improve the gut population?
Can you repeat that?
Where can we find the information to diet that could improve the gut population, the gut composition?
Well, I would say that if you read the literature, you'll get many opinions about what diet can do and can't do. Most of them, I think, are uninformed opinions based off of anecdote or empiricism or assumption. I think that in the future, we as academics and practitioners have to be accountable for what we say and recommend. We have to base our answers on true scientific evidence. And so it starts with us. We have to rebuild this culture, trust, reproducibility, and scientific rigor. I think it begins with us.
Okay, we have, if we don't have further questions from the floor, we have a few more questions from online. So one of the question is a technical ones. How the beneficial metabolites prebiotics are defined? Because on one hand, certain metabolites can act as inhibitors, suppressing the growth of pathogenic bacteria, while others can serve as carbon source, promoting the growth of beneficial bacteria. So those concepts of the good or bad metabolites are relative. And it is challenged to determine whether a particular metabolite is inherently beneficial or harmful. So supplementing with the metabolites may selectively promote the growth or harmful bacteria and disrupt the gut microbiotas. On the other hand, for other individuals, the same metabolite may enhance the growth. I think the audience was trying to give several examples whether some of the metabolites could be beneficial or harmful. So how do we choose that from the 20 markers out of 10,000?
Yeah, that's a very good question. I think that I don't know whether certain metabolites will be harmful or good, and it's going to be very individual, right? So, for example, too little of a short chain fatty acid probably will affect some patients in a way that is going to be more consequential than others. I speak, for example, in inflammatory bowel disease, where very typically fermentation is interrupted. You have very low levels of short chain fatty acids. This is like butyrate, acetate, propionate. These are very important in regulating the immune system as well as inflammation. And also they play a very important role in wound healing. So in that case, I think there's consensus that those metabolites are important to restore. In other cases, like TMAO, trimethylamine oxide, which Stan Hazen has shown to be associated with acceleration of cardiovascular disease. So you would use that as a marker for which there is a fair amount of human and experimental data that shows that TMAO it promotes inflammation, and it really accelerates atherosclerosis. So that would be a marker to include in the panel as a bad marker, right? So you want to make sure that your markers are representative of probably good and bad. But this has to be based off of scientific evidence, whether they're good or bad.
We got another questions from one of our colleagues who works on metabolomics. So the question is, in the 20 metabolites panel proposed, how many of them are endogenous metabolites? Are there any ways to tell whether the change in the abundance of these metabolites are due to microbes or the host?
Yeah, excellent question. So the answer is that we do have a way to sort of determine whether these are mostly microbe-derived or host-derived, but the point is well taken. Things like tryptophan metabolites can come from the host source. So when you're measuring it, it could be both host and microbe. However, if you look in a germ-free mouse, so these are mice that have no microbiomes, and you measure the metabolites, let's say tryptophan metabolites, what you're measuring is coming from the host. So you know that what component should be coming from the host versus from the microbe. Of the 20 that we've picked, almost all of them, with the exception of certain bile acids, are coming from the gut microbiome. Now, bile acids are very important for many, many reasons. But what happens is that the liver makes these bile acids. They're important for emulsification of dietary lipids, and it helps break down through the pancreatic enzymes, provides an interface for these enzymes to convert triglycerides to monoglycerides. That process is very much host dependent. But what we look at is conversion from conjugated to deconjugated bile acids, which clearly is a microbial function. We do not do that. We don't have the enzymes to do that. So that is definitely a microbial read. So we're not so much interested in individual bile acids. We're interested in the conjugated to unconjugated bile acid ratio.
Okay, thank you. Last questions. I think this question is from a research students. So as you mentioned, lifestyle such as diet affect human health through affecting on the gut microbiota and metabolites. Do you recommend direct intervention with gut microbiota or metabolites, or direct intervention with diet and gut microbiota or metabolites, just as a mechanism or index for improving the health?
So that's a great question, but a complicated one. And I'm going to go back to the defense that you have to consider ecological and environmental drivers that determine host microbe interaction. So again, the definition of a microbiome is not just the microbes, it's the ecosystem that the microbes are in, as well as the microbes. Okay, so if you consider that, the analogy is that whenever you think about host and microbe and whether you're going to restore, let's say, missing microbes, you have to think about what a colleague of mine calls seed and soil. Okay, seed is the microbes, let's say probiotics or FMT. So 99.9% of research and whatever commercial products are made are directed towards the seed. But if you don't have the right soil, the seed is never going to grow. So what do I mean by not the right kind of soil? If you have an inflamed environment, you're not going to engraft many normal or healthy microbes. They just don't have the properties to survive in that type of hostile environment. So what you have to do is you have to correct that ecosystem. Now, how do you do that? But there are many ways to do it. There are hydrogels, there are ways of making artificial mucus, trying to reduce the inflammatory response or immune activation. So there are things that you must do to restore the ecosystem so that when you put the seed back in, it will grow.
Okay. Due to limitation of time, we probably would end the QA section here. And thank you again for Professor Chen to give us a wonderful talk.
Thank you very much.
Well, thank you so much for your very wonderful lecture. I also wanted to thank all the audience for your presence here today, and especially for those who ask questions. I think your contribution to the lecture is very much appreciated. And this concludes today's lecture, and we are looking forward to your participation next time. Thank you very much.